me to be here. I like to focus my talk on the clastic rock record, which is a little bit under-investigated with respect to microbial structures. So I like to focus my talk on this here. So early life on Earth preserved in the clastic rock record. So first of all, I'm not going to talk about stromatolites here. I like to focus my talk on those structures that we call microbially induced sedimentary structures, abbreviated as MISS. So what are those structures? So first of all, we need, of course, a microbial mat, and I'm going to explain in a moment what exactly that is. It's an organic layer composed of microorganisms. That microbial mat is growing in a sedimentary setting, in a subaquatic sedimentary setting. Now, there are different types of sedimentary settings. One is a chemical setting, meaning there are minerals that precipitate. Think of a carbonate chemical setting or a setting where silica is forming. Microbial mats in such an area would form stromatolites. However, if we are in a clastic depositional area, we have physical sediment dynamics, and here the microbial mat has to interact with erosion and deposition and so forth, but there is no mineral precipitation. And the result are MISS. I like to show you a few examples for those structures. First of all, erosional remnants and pockets. That's an example from the modern, it's a tidal flat. Here you can see that this sedimentary surface is organized into two structural elements. The dark area here, the elevated area, that is a microbial mat. And here in between, you can see where the microbial mat was eroded away, and the sand that was originally beneath the microbial mat is now visible. It's exposed. Two more examples. The upper two photos show multi-directional ripple marks. On the left, we see a modern example. The scale is about 25 centimeters. You can see here a ripple mark area where the ripple marks have an orientation this way. Here is another area of ripple marks that have the same orientation, and here in the background are more. In between those areas, you can see ripple marks that run from left to right, so they have a different orientation. Such ripple mark patterns rise from the interaction of microbial mat growth in course of a year and the disruption by episodic storm events. On the right, we see a similar ripple mark pattern in the fossil record. That's from the 2.9 billion year old Pongola supergroup in South Africa. Here, the lower two photos show polygonal oscillation cracks. This is a view onto a modern microbial mat in Tunisia. Those mats can become very thick, about 4 centimeters. This here is a polygon here in the center. Here's another polygon, and here's another, and so forth. Those polygons are separated from each other by cracks. Within each of those cracks, a new microbial mat generation is forming, which is younger. I like to point out that each polygon, the margin of each polygon shows a ridge, a bulge. So here's one and here's one. So each of those cracks is indicated by two parallel running bulges. This is the same in the fossil record here. Here you see a polygon here, and here's another one, and here others. But you see very nicely those two ridges that run parallel. Another example are tufts. Here, a lateral view onto a modern microbial mat. Sometimes microbial mat surfaces show those tufts, which are formed of microbial filaments that migrate along each other to form those elevations. They're very small, just 1 to 2 millimeters. This is a lateral view, and this is a view top down. So on the right, we see a modern microbial mat that has those tufts forming where those little ridges are meeting. Those ridges here form a honeycomb pattern, and if we do statistical analysis, we can compare those patterns with fossil ones, and so we know it might be the same. This is an example here for fossil microbial mat from the 3.48 billion years old dresser formation in the Pilbara. I will talk about this formation in the second part of my talk once more. Microbial mat chips are fragments of microbial mat. This is a modern example here. Very typical are those embayments and lobes of those mat chips. So those chips have a different morphology than mud chips, M-U-D, mud chips. So mat, a microbial mat chip here. This is a beautiful example preserved in the dresser formation, where we even can see different layers of microbial mat. So note again, here we have this lobe, here's another lobe, here's an embayment, and so forth. So microbial mat chips, this here is 1 centimeter for scale. Now, if microbial mat chips are being transported, they can be rounded. Here, beautiful examples from the dresser formation again, and here, modern counterparts. So how do those structures form? This is a view into a modern sandy tidal flat along the North Sea coast, and we get the impression that those sediments are composed entirely of sand grains. However, if we take a magnifier, we see that those sediments are actually widely colonized by a great variety of benthic microorganisms. And of those, cyanobacteria play a major role with respect to sedimentary structures that are being formed. Those are 2 examples of cyanobacteria. This one here is Merismopedia punctata. It forms pillow-like clusters of spheroidal cells, here attached with the extracellular polymeric mucilages, which are adhesive slimes to a hard substrate. This is Microcoelius tonoplastis, which forms elongated bundles of filaments. Cyanobacteria are photoautotroph, and they can become pretty big. So that's why the older literature calls them actually blue-green algae. If cyanobacteria start to colonize a sediment, they initially form so-termed biofilms around each individual grain. The biofilms are composed of the single cells themselves plus their EPS, those mucilages. If such biofilms continue to grow over several months, in the end, a microbial mat is forming, which you can see here on the left. Here's a knife for scale. Microbial mat are very, very coherent. So this is really an organic layer, almost entirely composed of microorganisms. Those layers are very coherent. So you literally can bend down, pull it off the sedimentary surface, roll it up like a carpet, put it on the shoulder, and walk away with it if you want. And clearly, such microbial mats must have an impact onto sediment dynamics, on erosion, and so forth. So the next question I have is, how do prokaryotes interact with physical sediment dynamics in order to survive? So microorganisms obviously are very, very small. And if they live in between grains that have relatively the size of boulders, it's a good idea to have some survival skills. So how do microorganisms, in this case cyanobacteria, react to erosion and deposition of sediment? This is shown in this graph here. It's composed of 3 arrows. This arrow here shows intensity of erosion. This arrow here shows intensity of deposition. And this here is increase of latency or stasis. That's a time period during which the sedimentary surface is not disturbed by erosion or deposition. Those rectangles here show individual cyanobacteria populations and their response to specific dynamic conditions. So let's take a look at this here. What happens if the sedimentary surface is completely quiet? No sedimentary grains are moved around. What we can see are two things. First is binding. This is a close-up view onto a fossil microbial mat. You see here beautifully the filaments entangled, forming this meshwork, which is typical for a microbial mat. Now, such a meshwork has to be formed first. And this formation of such a meshwork, this is what we call binding. The microorganisms move actively through the sediment. They communicate with each other through cell-cell communication and quorum sensing and so forth. And they establish this type of meshwork where each individual filament has a position optimal with respect to its neighbors, so that the metabolic activities can interlock, so they can all work together to process energy. Now, if the sedimentary surface remains long enough calm, growth also takes place by cell replication, EPS production. Here you see a vertical section through a sediment. Here are ripple marks. And the ripples are completely covered by a microbial mat that's almost a centimeter thick. It grows very thick, specifically here in the ripple valleys. Now, with respect to the situation of a microbial mat, like here in this example in a tidal flat, different types of colonization, different types of microbial mat form. We can have just biofilms that form around individual grains. That is the case in the lower intertidal zone, where the sedimentary surface is constantly reworked. So no real microbial mat can form. In the upper intertidal zone, we may have endobenthic microbial mats, which are microbial mats that live inside the sediment in the uppermost millimeters. So here you see the ripple marks of that surface, but it's covered, it's interwoven by a microbial mat. In the lower supratidal zone, where we have long periods of calm conditions, epibenthic microbial mats can form that really cover a sedimentary surface like a tablecloth, and those are the microbial mats that we are familiar with from textbooks. So what happens during erosion? This is a lateral view onto, there it is, onto a microbial mat here in the lab. So this is the microbial mat surface here. We see that the filaments are interweaving the sedimentary grains, and also that there is a lot of those mucilages, those glass-like EPS that the microorganisms secrete. This arrow here shows there is a current that crosses the sedimentary surface. We did analyses with different types of microbial mat in the field and so forth. And there are different values of biostabilization, of sediment fixation of those microbial mats, meaning the resistance of the sedimentary surface against erosion is increased by microbial mats. What happens during deposition of sediment? So here, this is the same microbial mat. You can see sedimentary grains that rain down onto the microbial mat surface. As a response, the microorganisms orientate perpendicular to the microbial mat surface. They reach up into the seawater and they trap and bind sedimentary grains and incorporate them into the developing microbial mat fabrics over time. Now, if we have this mechanical interaction of microbial mats with deposition, with erosion, or with stasis or latency, we have specific sedimentary structures that document this interaction. It's almost like traces, and I'm getting back to that once more in a moment. This graph here shows different MISS in relation to the main microbial parameter that leads to the formation, like biostabilization is here, baffling trapping is here, binding down here, and growth up here. Those MISS in the center actually rise from an overlap of all 4 microbial activities. MISS are known from the entire fossil record. They occur in clastic sediments since the early Archean until today. And you can see we know them from deltas, from shelf settings, lakes, playas, and so forth. So interestingly, we can study their formation in the modern to have data to understand the formation and to also understand evolution of prokaryotes in course of Earth history. So how are MISS preserved? So we move now away from formation towards preservation, which is a second step necessary if we like to find fossil MISS. First of all, we have to understand that MISS have a threefold character, and that helps us as geologists to map for those structures. First of all, they have the character of a sedimentary structure because we see macroscopic structures in the field, either in the modern or in the rock record. We can also regard MISS as traces or trace fossils, because we have, as a cause of those structures, the interaction of organisms with a sediment. And we also see, as you will see in a moment, we have body fossils of microorganisms. So this is just an example for a macroscopic structure. This is an MISS. You've seen this before, the microbial mat chip, clearly visible in the field. But MISS have trace fossil character. Think of a worm that is moving through sand. This is Arenicola marina, very common along the North Sea coast. So this worm is moving through the sediment and it's causing a trace. This is a fossil trace here in sandstone, and very similar. We can imagine if an entire microbial mat is moving with ripple marks and so forth and interacting with the sediment, it causes a trace. So it is a trace fossil if we find something like that in the fossil record. But we also have body fossils. This here is a piece of a rock from a macroscopic MISS. We can cut those rocks into very thin slices, mount them on a glass slide, and then investigate the mineralogy that is composing the sedimentary structure. This is basically a microscopic here, and what we see are fossil filaments, fossil bacteria, sometimes quite nicely preserved. So here are a few examples. This here is a thin section view of a modern microbial mat. You see sedimentary grains embedded in this microbial mat fabrics here. Those are filaments here, those dark lines. This more whitish material in between are those EPS, those extracellular polymeric substances. And here you see something very similar in the fossil record. That is a thin section of a rock. I believe this is from the Pongola supergroup. Here you see a grain, sedimentary grain. Here's another one. But clearly you see those beautiful body fossils of microorganisms as well. With respect to mineralogy, those filaments are preserved by carbon, pyrite, hematite, chemosite, and then also the EPS are preserved as chert. So that is a mineral association that we can see quite frequently. Let's move indeed into the Dresser Formation, which has an age of 3.48 billion years. The Dresser Formation is a rock succession of early Archean age that occurs as part of a tectonic structure that's called Norfolk Dome, situated in the Pilbara block in Western Australia. The Dresser Formation forms part of the very, very famous Warrawoona group, where a lot of people have detected microbial fossils, stromatolites. There's a lot of geochemical analysis done and so forth. So part of this Warrawoona group is the Dresser Formation with an age of 3.48 billion years. This says 9, but that's a typo. This is an example for one of the rock successions that we were mapping millimeter by millimeter. It's a clastic succession and some carbonate in there as well, and it is silicified, meaning very early on silica precipitated because the sediments were affected by volcanic activity. Here we have a lot of MISS. Now, if we reconstruct the paleo environment, we can show that there was an ocean. The ocean was separated from the land by a very shallow shoal, which I'll show you in a moment, which is carbonate ooit. And then we have a tidal flat, where some areas were subaerially exposed. We have a lot of evaporite forming, like gypsum and so forth. And then there are sandy areas with tidal channels and tidal pools. And in those channels, we see some stromatolites. Now, as a modern example, more or less, we can use a tidal septa like this one here in Tunisia, where we see tidal channels here filled with water. Then there are areas here in the front that are subaerially exposed most of the time. And then the actual intertidal here, where we have different types of microbial mat growing. So let's take a look. The subtidal zone, which includes the tidal channels, show stromatolites, which are not MISS, but I like to mention them just because they are there. But we also see in those sandstones that we have ripple marks there that are covered by biofilm. So this is the top of a ripple mark, so ripple mark crest. This is the ripple slope. This is the ripple valley here. And this black line here, this is a fossil biofilm, a very thin microbial mat that was draping this ripple mark and made sure that the ripple mark is basically frozen. It does not move anymore because it's biostabilized. Here on this side, you see how this ripple valley was filled in step by sediment. And each time the current was receding, a biofilm was forming. So we have different biofilm layers here. Little microbial mats forming here. This is a close-up. And here are individual little tufts still nicely preserved. Now we investigated this fossil microbial mat here. So this is a little tuft here. Here's another one, and found pyrite and carbon and other mineralic substance. But this carbon, pyrite, and so forth clearly have a biological signal. So this indeed reflects the original carbon that was composing the microbial mats. Now let's move up into the intertidal zone. I showed you before erosional remnants and pockets. This is a close-up here. So we look down onto a microbial mat surface. This is the microbial mat here in the modern. This is the edge of the microbial mat. It's a little bit fringed because of erosion. Here's a little fragment of microbial mat that was originally situated here, but then it was ripped off and redeposited right away. This is another microbial mat chip. And now take a look at this beautiful fossil example here. This is a fossil microbial mat. Again, we look down onto the surface. This is the edge of the microbial mat here, where it was eroded away And here is a little microbial chip that was ripped off 3.5 billion years ago and is still in situ, beautifully preserved. Here's another one up here. If we do a thin section of material like that, we see fossil filaments. Here is a modern example similar to the one that you have just seen. Very typical for the early Archean is that carbonaceous matter is shriveling up, it's shrinking, it forms little lumps of carbon, but still we see the original shape of the filaments is preserved. Now, we also here did our chemical analysis, we see that, again, there's carbon, and this carbon has an organic signal. In the lower supratidal zone, now those are areas that are more exposed subaerially for a longer time. We have signs of polygonal oscillation cracks. So here you see a polygon, here's another one. Here you see those, those cracks that are overgrown by younger microbial mat, and here is the same thing. Each polygon in this fossil example has a hole in the center, which is typical for such polygonal oscillation cracks. If a microbial mat is drying out, gases assemble underneath the mat, and each polygon is domed upward. And eventually in the center of the top, the roof of the dome, the microbial mat ruptures and the gases escape and it smells impolitely. But then the microbial mat sinks down again, but the hole in the center remains. And this is what is recorded on this example here. Then there are also lagoonal areas that have a pattern of black-white layering. We compare that with biolaminites. Here's a modern example. This is a vertical cut through a microbial mat, a modern one about 4 or 5 centimeters thick. We see dark layers here, that's microbial mat, and in between those lighter colored layers, which is just sediment that was deposited onto microbial mat. So this biolaminite grows upward in course of time. If we take a look at thin sections, we see in those biolaminites microbial mat chips or little roll-ups here, chips that are rolled up, very nicely preserved. And of course, they have, again, this typical biological signal. So conclusions for the dresser formation are, we know that the dresser is one of the oldest non-metamorphosed sedimentary units, meaning the rocks have not experienced so much alteration by hot water that's circulating through the rock or by tectonic faulting and folding and so forth. So we still see the original sedimentary rocks. We interpret the rock successions we studied as an ancient tidal subcar, very similar to modern tidal subcars. And we have MISS morphologies that correspond to those morphologies that appear in modern subcars as well, specifically in the same settings. And we concluded at that time that fossil and modern MISS are formed by organisms that reacted in very similar ways. So we suggested there were cyanobacteria around already. So that was an overview about MISS in general. I've written a book. If you are interested, you can borrow it. You don't need to buy it. This is a book that summarizes the field a little bit. It's somewhat older by now. It's from 2010. Now, since a lot of you are astrobiologists, I wrote an article which is an introductory article into MISS and the implication for Mars exploration. That article came out 2021 in Astrobiology. It's open access. And this is written, so I hope, so for the general public to understand. So it's very introductory. And with that, I would like to thank you very much for your attention.